Start, sir.
Okay, sir. Thank you. Good morning, students. This is last session of cell biology. As usually, this is appreciation time. Ankita and Prasuna from TSRJC Vangara. Kirtana and Sankirtana from TSRJC Vaira. Neelima and Srija from TSRJC Chautuppal. Harita and Ramadevi from TSRJC Balnagar. Surya Prakash Reddy and Yashwant from TSRJC Beechupally. Congratulations. So once we have to recap phases of cell cycle. So this is phases of cell cycle. Already previous class we are learned about different stages of cell cycle. Cells are divided once in approximately every 24 hours. Cells are divided once in approximately every 24 hours. The cell cycle is divided into two basic phases. Cell cycle is mainly divided into two basic phases. One is interphase, one more is M phase. So one is interphase, second one is M phase. Interphase is resting phase. M phase is that is dividing phase. So M phase is mainly takes place in two subphases. One is karyokinesis, second one is cytokinesis. One is karyokinesis, second one is cytokinesis. Karyokinesis is nothing but nuclear division. Nucleus is divided into two equal halves. That is karyokinesis. In case of cytoplasmic division or cytokinesis, after nuclear division, cytoplasm also will divide it into two equal halves. That is cytokinesis or cytoplasmic division. So, in case of interphase, again subcategorized into three subphases: G1 phase, G1 phase, next S phase, and Z2 phase. G1, S, and Z2 phase. G1 phase is a gap 1 phase. G2 is a gap 2 phase. S phase is a synthetic phase or synthesis phase. G1, G1, S, and G2. G1 phase followed by S phase, S phase followed by Z2 phase. Nothing but three phases are there in interphase: G1, S, and S2 phase. First of all, G1 phase or gap 1 phase. G1 phase is mainly cell is metabolically active for it is ready to divide. Cell is metabolically active in this phase, that is G1 phase. Next, cell is metabolically active but does not replicate DNA. DNA is not replicate or DNA is not double. So DNA is not replicate. Only cell is metabolically active or ready to cell division. Next, S phase. S phase, which is also called as synthetic phase. S phase, it is also called as synthetic phase. DNA synthesis or replication is takes place in this phase. In S phase, what will happen? One is DNA synthesis or DNA replication is takes place. Amount of DNA per cell is double. So one copy of DNA is further divided into two halves. I mean, two sets. One set of DNA changes into two sets. Means DNA amount is double. DNA is replicated. One DNA copies another one. So here denoted that 2 copy of DNA then increases into 4 copy of DNA. There is no increase the amount of chromosome number. Only DNA mainly present in inside of the chromosome. DNA present in inside of the chromosome. So inside of the chromosome, DNA is replicated and it is changes into double. But there is no changes in chromosome number. Chromosome number is as usually same in this phase. This is gap 2 phase. This is gap 2 phase. Gap 2 phase mainly responsible for proteins are synthesized in preparation of mitosis while cell growth is continued. So proteins are synthesized. What are the proteins are required for cell division? That proteins are mainly synthesized by the cell in G2 phase. One thing, it is preparation for mitosis while cell growth is continue. Cell growth is usually continue. And one more change is proteins are synthesized for cell division. What are the proteins are required for cell division? That proteins are synthesized by in the phase that is G2 phase. So here G1 phase is a growth phase. G1 phase is a growth phase. Next, S phase is a DNA synthesis takes place. It means DNA, one copy of DNA changes into two copy DNA. Next, G2 phase, it is growth phase and it is preparation for mitosis. It is ready for mitosis or cell division. And last, M phase. M phase is mitosis or cell division phase. M phase is that is a division phase, either mitotic division or meiotic division. G1 is growth. S phase is for responsible for DNA synthesis. G2 is responsible for cell growth and preparation of proteins for what are the proteins are required for the cell division. And fourth phase, that is M phase. This M phase mainly responsible for that is the cell division, either mitosis and meiosis. Next, M phase. Next, M phase. So M phase or this is division phase mainly followed by two steps. Mainly followed by two steps. One is karyokinesis. Karyokinesis is nothing but nuclear division. Nucleus is divided into two equal halves. This karyokinesis is followed by cytokinesis. Cytokinesis means cytoplasmic division. After nuclear division, cytoplasm also will divide. So that is cytokinesis. So cytokinesis again categorized into four phases. Cytokinesis again categorized into four stages, four phases. One is prophase, metaphase, anaphase, and telophase. Cytokaryokinesis, nothing but nuclear division. Nucleus is divided into two equal halves by these four stages. One is prophase, metaphase, anaphase, and telophase. These four stages already we are discussed already in our previous class. So this is mitosis or equational division. Why? Because it is equals two daughter cells are equals to similar to parent cell. That's why it is equational division. Equational division means two cells. What are the daughter cells? Two are formed here. One here. These two daughter cells are they are similar to parent cell. Similar to parent. That's why it is a equational division. This is prophase, metaphase, anaphase, and telophase is cytokinesis. These four stages already we are discussed in our previous class. So here, this is our, so in mitosis, early prophase, this is early prophase. In early prophase mainly, chromosomes are mainly, they are thread-like structures. Thread-like structures means chromatin material. This is we are observed in early prophase. In late prophase, after late prophase, so we are observed this what is it? Chromatid material. Chromatin material it changes into chromosomes. Chromatin material changes into chromosomes. So before stage, early stage, that is chromatin material is there. That chromatin material changes into chromosomes in prophase. Next, metaphase. Next, metaphase. In metaphase, what will happen? That is the centriole starts formation of spindle fibers. That spindle fibers generally attaches to kinetochore of the cell, kinetochore of the chromosomes. So this is a centriole, here one centriole, here one centriole. This centrioles moves to opposite pole, here this one, here this one, opposite pole. Then after this centriole starts to formation of spindle fibers. This thread-like structures is called spindle fibers. That spindle fibers attaches to here particular the place that is kinetochore, attaches to kinetochore. Then after this is metaphase, actual metaphase. In this actual metaphase, what will happen? Centrioles, there formation of spindle fibers attaches to centromere or kinetochore. Then after completely still attaches to centromere in this phase, metaphase. And then anaphase. In this anaphase, what will happen? These spindle fibers moves to, opposite poles. They are pressure, generate exact some pressure towards the chromos, kinetochore or centromere. Centromere is break down into two equal halves. Then after, half of the chromosomes moves to opposite pole and half of the chromosomes moves to another opposite poles. So what will happen? Centrioles, what is it? Spindle fibers starts the movement. Due to this movement, kinetochore or centromere break down into two halves. Then after, these chromatids are moved to opposite poles. Here we are observed. In telophase, what will happen? The centrioles moves to opposite poles. Then after, nuclear material, genetical material is totally completely becomes to half number. This genetical material contain half number of chromosomes. The number of half number of chromosomes into opposite poles. Completely, nucleus is divided here. In telophase, nucleus is divided. Then after, nucleus is completely divided, but cytoplasm not. Sorry, then cytoplasm is divided during the cytokinesis phase. Cytokinesis, what will happen? Cytokinesis takes place after telophase. A result of two daughter cells are formed. We are starting with only one cell, and we are taking two cells, two daughter cells. These two daughter cells are similar to their parent, that is equational division. Then after, these two daughter cells are similar, their genetical material is present in parent. Similar genetical material and similar genetical cytoplasm. So this is mitosis. Significance of mitosis. Next, what is importance of mitosis? So production of daughter cells is one thing. From one parent cell changes into two daughter cells. These two daughter cells they are similar the genetical material as usually parent. So formation of two daughter cells with identically similar to their genetical material as per parent. The growth of multicellular organism due to mitosis. The growth of the plants mainly due to mitotic division, because number of cells are produced from the one cell to another cell. One cell replicate into two cells are double into four, like that. Many cells are formed by this stage, that is mitosis. Due to this reason, the growth is takes place in plant body. Next, cell growth distribution of equal amount of the nucleus and cytoplasm. So here we are observed, cell growth is result distribution of distribution ration of nucleus and cytoplasm. Equal amount of the cytoplasm enters into daughter cell. Equal amount of the nucleus also enters into daughter cell. So therefore becomes to essential for the cell restore the nucleocytoplasmic ratio. Restore means completely that daughter cells they are obtain cytoplasm and nucleus. The equal amount of cytoplasm, equal amount of nucleus they are obtained from the parent cell. Next, the mitosis is very plays significant role in cell repair. Cell repair. When old cells are damaged, the new cells are formed from the pre-existing cells. That is cell repair. Cell repair is mainly done by, that is, mitosis. Cells upper layer of the epidermis and lining of gut in our digestive system, in blood vessels also continuously replaced. So by the help of mitotic division, epidermal cells, lining of gut cells and blood cells, they are divided, continuously divided from new cells, continuously dividing and formation of new cells. So mitotic division is the meristematic tissue and lateral cambium result continuous growth plants through their lifetime. In plant body, meristematic tissue, and lateral cambium, they are mainly continuously growth through their lifelong. It means meristematic tissue in meristematic cells, we are observed it is continuously dividing cells in the throughout the lifetime. So meristematic cell shows continuous division throughout the lifetime. Next, meiosis. So this is a meiosis, second type of cell division. We are observed different stages: prophase, metaphase, anaphase, and telophase. By the result of mitosis, 2 daughter cells are formed, but result of meiosis, 4 daughter cells are formed. This is meiosis 1. During the meiosis, what will happen? 4 stages are there: prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. First of all, what will happen in prophase? Already we are discussed. Prophase again categorized into 5 different stages: leptotin, zygotin, pachytin, diplotin, and diakinesis. In prophase, one of leptotin, condensation of the chromosomes is one thing in leptotin stage, and zygotin phase, homologous chromosome starts to pairing, that is synapsis, second stage. And third stage, pachytin stage. In this pachytin stage, what will happen? Crossing over takes place between the non-sister chromatids. Non-sister chromatids means this is a blue one and red one. That is non-sister chromatids that exchanges the genetical material by the crossing over. Diplotin is mainly repulsion takes place, want to go separate. So both homologous chromosomes, non-sister chromosomes, they are bind together. That is synapse. Then after they are to go separate. Already in crossing over, exchange the genetical material. So DNA of the red one enters into blue one. Blue one of the genetical material there enters into, that is red one. So in here we are observed one paternal and maternal chromosomes like that. Next, diakinesis is last stage. So in the diakinesis, what will happen? That homologous chromosomes are want to separate, but still remaining attaches at the tip. Only attaches to at the tip. Already we are discussing yesterday. This is prophase. Next, metaphase. What will happen in metaphase? Chromosomes are arranged on equatorial plate. So these chromosomes are arranged on equatorial. This is equatorial plate. Here one chromosomes, here one set of chromosomes, here one set of chromosomes. Just we are observed. Equatorial plate is at middle. Equatorial plate is middle. So this is equatorial plate. Equatorial plate or metaphase plate. What we observe, the chromosomes are there arranged on equatorial plate. Next, anaphase. This is anaphase. What will happen? Half number of chromosomes moves to opposite poles. This half number of chromosomes moves to one pole. Half number of chromosomes moves to another pole. And telophase, what will have? What we observe? That is nuclear membrane reappeared, nucleolus reappeared, and all type of cell organs again reformed. And what will happen? Spindle fibers are disappeared. This is here. Metaphase and anaphase we are observed spindle fibers. This is spindle fiber. This is spindle fiber attaches to kinetochore. But in this telophase, what we observe? Spindle fibers are absent. Spindle fibers are disappeared. Then after chromosomes recoils to form chromatin material. Chromatin material means these chromosomes to contain mainly changes into that is chromatin material at the end of telophase. Result of this meiosis 1, what will happen? Two daughter cells are we get. Two daughter cells are formed. Here one daughter cell, here one, this is second daughter cell. From the one cell to two daughter cells are formed by the meiosis 1. Common thing is the prophase 1 again categorized to 5 categories: leptotin, zygotin, packaging, diplotin, and diakinesis. So these 5 stages already discussed. Then after metaphase, anaphase, telophase. A result of telophase after cytokinesis takes place. Cytokinesis 1 is takes place. Result of cytokinesis 1, two daughter cells are formed completely. Then after this two daughter cells, they contain not chromatids or chromosome. They are chromosome, the material they are recoils, that changes into chromatin material. Chromosomes are changes into chromatin material. This is stages of meiosis 2. Stages of meiosis 2. So already in meiosis 1, two daughter cells are formed. We know that this is one daughter cell, this is another daughter cell. This is first daughter cell, this is second daughter cell. In meiosis 1, two daughter cells are formed. This is one daughter cell, this is another daughter cell, two daughter cells. After cytokinesis 1 of meiosis 1, the cell is going to be resting phase, that is interphase. Interphase nothing but G1, S and G2 phases. Then after start, they are enters into prophase 2 or meiosis 2. In the meiosis 2, what will happen, sir? The meiosis 2 again categorized into four stages, main stages, and the fifth one, that is cytokinesis 2. In meiosis 2, prophase 2, metaphase 2, anaphase 2, and telophase 2. So this is prophase 2, metaphase 2, and anaphase 2, and this is telophase 2. After telophase, from one cell to two daughter cells are formed as usually. And what are the changes we are observed in these stages? Once we have to recap. So first, what are the changes that takes place in this first cell? Second changes also takes place as usually in second cell also. What I'm saying, this is first cell. What are the changes that takes place in first cell? Second cell also they have same type of changes. First cell formation of two haploid daughter cells, and second cell formation of two haploid daughter cells. Result of two cells, four cells are formed. From the diploid cell to haploid, four daughter cells are formed. So prophase 2, first phase, what will happen? Chromosomes becomes to condense, nuclear membrane and Golgi complex and endoplasmic reticulum are de-appeared. What will happen? Chromosomes becomes to condense. Before stage, chromatin material is there, but here we are observing chromosomes. This process is done by chromosomes becomes to condensation process. By the condensation process, that chromatin material changes into chromosomes. And in first case, what will happen? Endoplasmic reticulum and Golgi apparatus and nuclear membrane is disappeared. Nuclear membrane, Golgi complex, endoplasmic reticulum are all cell organs. They are going to be not appeared in this phase, first phase. In second phase, that is metaphase 2. In metaphase 2, what will observe? Chromosomes are connected to centrioles at kinetochore. This is one centriole, this is one centriole. Two centrioles are connected to, this is kinetochore or centromere. Centromere. This is kinetochore. At middle one, centromere. Next, in metaphase, what will happen? Chromosomes, they are aligned at equatorial plate. Already we said this is equatorial plate at middle plate. This is equatorial plate. Chromosomes are arranged on equatorial plate, but in prophase 2, what will observe? Chromosomes are irregular. Their movement is irregular. But in metaphase 2, what will happen? The chromosomes are moved to on the equatorial plate or metaphase plate. Next, anaphase 2. This is anaphase 2. Splitting of centromere of each chromosome. That is, centromere attaches to here. This centromere is break into two equal halves. That's why centromere of each chromosome is splitting or break down into two equal halves. Result of chromosomes moves to opposite poles. So this half number of chromosomes moves to upside, and half of the chromosomes moves to downside. Here also same. Half of the chromosomes move to one side, half of the chromosomes moves to another side. Next, at the telophase is the last phase. Nuclear membrane again reformed. All cell organs also reformed, and a result of telophase, finally 4 daughter haploid cells are formed. This is 1, this is 2, 3, 4. These 4 haploid daughter cells are formed by the end of telophase. Then after cytokinesis, completely we are observed 4 different cells are formed by the cytokinesis. This is karyokinesis. Then after cytoplasm also will divide from this. This is karyokinesis. Then after cytoplasm also will divide here also, here also. So cytoplasm also divide to formation of 4 haploid daughter cells. This is meiosis 2. Next we are observed importance of meiosis 2. Significance of meiosis 2. So conservation of specific chromosome number. One thing is conservation of specific chromosome number of each species achieved across the generations sexually reproducing organisms. What are the organisms? They are reproduced there by the sexual method. Their chromosomes, specific chromosomes, they are mainly conservationist takes place. It means chromosomes from the one parent and chromosomes from the another parent, that genetical material transported to the daughter cells or daughter organisms. Main thing is chromosomes, they are genetical material of the parents. One of the copy is enters into daughter cells. Next, even through the process for paradoxically result reduction of chromosome number is half. In case of observed mitosis, 2 daughter cells are formed from the one parent, diploid parent. Changes again diploid number of chromosomes were observed. But in meiosis, starts with the diploid parent, but ends with the haploid daughter cells. So that's why the chromosome number is becomes to half. Chromosome number becomes to half. So this meiosis takes place in sperm mother cell and ovum mother cell in human, like that organisms, animals. In case of plants, pollen mother cell and ovum mother cell in, that is the in case of plants. So it is also increase the genetical variability in the population. How it is possible? Genetical variability. Genetical variability means genetically modified cells are formed. It means some of the characters are we are obtaining from the parent cell, one parent. Some of the characters we are getting from another parent. So this is genetical variability. Both type of characters are we are seen in, we are observed in daughter cell. So the daughter cell contain genetical variability. So half of the chromosomes from the one parent, half of the chromosomes from the another parent or another plant. So one parent characters and second parent characters, two types of plant characters were observed in daughter plants. So it is a genetical variability. Variations we are observing, that is genes, that is observed in organisms. So these variability or genetical changes, they transfer from one generation to another generation. Number of generations, this genetical variability is passing to over generations. So variants are very important for evolution. Variations are very important for this evolution. It means different types of variation in organism by the DNA, it is better to survive in the world, better to survive in any situations. That is evolution. Next, this is cell structure and functions were observed. Next, assignments. So I will give assignments now. Describe the cell organelles with different type of cell organelles with help of diagrams. So first question, describe the all cell organelles with the help of diagrams. You better to write each and cell organelle name and their structure and functions, then after one diagram. So next question, explain about different types of carbohydrates in detail. Carbohydrates are mainly different types is there. I mean monosaccharides, disaccharides, oligosaccharides and polysaccharides. So different types of carbohydrates. You better to write different types of carbohydrates in detail, their structure and their function. So next, third question, what are the various stages of meiotic phases 1 and meiotic prophase 1? In case of I'm asking, in meiosis 1 of prophase 1, what we have what will observe? 5 stages will observe: leptotin, zygotin, pachytin, and diplotin diakinesis. So these 5 stages you have to mention here. So what are the various stages of meiotic prophase 1? Enumerate the chromosomal events during the each stage. What are the changes we are observed in chromosome? You better to write. Next, explain about fluid mosaic model of plasma membrane. In case of plasma membrane, we are observed fluid mosaic model. Fluid mosaic model. It means proteins, lipids, and carbohydrates. Lipids are bilayer, nothing but 2 layers: hydrophobic tail, hydrophilic head. We observed phospholipid bilayer, then intrinsic proteins, extrinsic protein, glycoproteins. So these are the fluid mosaic model proposed by Singer and Nicholson. I'm already explained in our previous classes. Once we have to recap and then have to write plasma membrane structure and functions. So these are the 4 different what is that questions. I will explain one more time. That's what is that assignments. First question, describe the all cell organelles with the help of diagrams. You better to explain each and every cell organ with the help of one single diagram. Then after, second question, explain about different type of carbohydrates in detail. Explain about different types of carbohydrates, nothing but monosaccharides, disaccharides, oligosaccharides, and polysaccharides. So this is next. What are the various stages of meiotic division in prophase 1? In prophase 1, we are observed 5 substages. What is different type of stages? Leptotin, zygotin, pachytin, and diplotin, and diakinesis. Okay, so this is about cell structure and function. Only one chapter is there. Only one chapter is remaining. That is plant ecology. From tomorrow's session, they will start plant ecology. So until today, all chapters are completed except plant ecology. This chapter is mainly cell structure and functions. In cell structure and functions, we are observed different type of cell organelles. Cell is the basic unit of life. Second chapter, biomolecules, and third chapter, that is cell cycle and cell division. So today class 1 we have to recapitulate. So phases of cell cycle, main phases are interphase and M phase. So M phase mainly again categorized into two phases, karyokinesis and cytokinesis. And interphase again three types, G1, S and G2 phases. G1 phase mainly metabolically active the cell, but DNA is not replicate. In S phase, what will happen? Synthesis phase, it is synthetic phase. DNA synthesis is takes place and DNA replication also takes place. It means DNA becomes to double. Amount of DNA per cell is double. 2 copy of DNA then changes into 4 copy of DNA. There is no increase the chromosome number. In G2 phase, what will you observe? What are the proteins are required for the cell division? That proteins are mainly synthesized in G2 phase. So M phase. M phase mainly karyokinesis and cytokinesis. Karyokinesis means nuclear division. Cytokinesis means cytoplasmic division. Karyokinesis again categorized into 4 substages: prophase, metaphase, anaphase, and telophase. This is mitotic division, mitosis. This is equational division because parent cell, daughter cells are similar to parent cell. This early prophase, the chromatin material changes into, that is, chromosomes. Metaphase, the centrioles are attached to kinetochore, and in metaphase what will happen? Centrioles are attached. What is that? Spindle fibers are attached to, that is a centromere. Anaphase, half of the chromosomes moves to opposite poles. Half of the chromosomes move to one pole, and remaining half of the chromosomes moves to another pole. And result of telophase, genetical material becomes to half. Genetical material be totally arranged in two sets. I mean, half of the material moves to opposite, and another half of the genetical material or nucleus moves to opposite pole. Two daughter cells are formed by the help of cytokinesis. This is significance of the mitosis. Mainly two diploid daughter cells are formed. Mainly it is involved in distribution of equal amount of the nucleus in equal amount of the cytoplasm to daughter cells. And mitosis is very helpful to body repairing. This is meiosis, prophase 1, metaphase 1, anaphase 1, and telophase 1. In prophase 1, again categorized 5 substages: leptotin, zygotin, packaging, diplotin, diakinesis. In case of leptotin, condensation of genetic material takes place. Zygotin, homologous chromosomes starts pairing, that is synapsis. Packaging, crossing over takes place between non-sister chromatids. Non-sister chromatids means blue color one and red color one. These two are non-sister chromatids, that is both are connected, that is synapses, that is crossing over. By this crossing over, genetic material is exchanged between paternal and maternal chromosomes. Next, what is that? Packed in crossing over takes place, and diplotin, repulsion. Repulsion nothing but want to go separate. The both chromatids go to separate, move to separate. Then diakinesis is mainly the tip of the non-sister chromatids still remaining attaches. Next, metaphase 1, chromosomes are arranged on the equatorial plate. And anaphase 1, what will happen? Number of chromosomes moves to opposite poles. I mean, half number of chromosomes moves to opposite poles. And telophase 1, nuclear membrane and nucleolus and spindle fibers. I mean, nuclear membrane, nucleolus are reappeared, but spindle fibers are disappeared. Result of meiotic 1, meiosis 1, two daughter cells are formed. After these two daughter cells are involved in, that is meiosis 2. Meiosis 2 starts with prophase 2. So prophase 2, what will happen? Chromosomes becomes to condensation, and nuclear membrane, Golgi complex, and endoplasmic reticulum, they are becomes to disappeared. And metaphase 2, what will happen? In meiosis 2, chromosomes are connected to centrioles and at the place of kinetochore. Chromosomes get aligned on equatorial plate or metaphase plate. In case of anaphase 1, what will happen? Splitting of centromere of each chromosome. Chromosomes are moved to opposite poles, and telophase is the final phase. After moving of the chromosomes, then after nuclear membrane again formed. So In the phase of telophase, nuclear membrane is remaining, reformed. Cell organs also reformed, and centrioles are what is that? Spindle fibers are absent. So, result of this telophase 2, 4 haploid daughter cells are formed. Here, diploid daughter cells are formed in prophase 2. And end of the telophase 2, we are observed haploid daughter cells. This significance, conservation of the genetic material from the parents to offsprings from the one plant to another plant. Daughter plants are similar to that is a parent plant. So main thing is reduction of chromosome number. In case of mitosis 1, we are observed diploid chromosomes observed in parent and diploid chromosomes observed in daughter cells also. But meiosis division, diploid mother cell and changes into haploid 4 daughter cells. So this is the assignments. Better to write different type of cell organs with the help of diagram, different type of carbohydrates, and stages of prophase 1 in meiotic division, and explain about fluid mosaic model of plasma membrane. So in this unit, cell structure and functions, we are observed 3 chapters. Cell is the basic unit of life. We are observed different type of cell organelles and their structure and their function also. And in case of biomolecules, we are observed different macromolecules and micromolecules. Macromolecules, carbohydrates, proteins, and fats. Carbohydrates are mainly monosaccharides, disaccharides, oligosaccharides, and polysaccharides. In case of proteins, essential and non-essential proteins and their structure, primary structure, secondary structure, tertiary structure, and quaternary structure. And lipids. Lipids are mainly simple lipids, conjugated lipids, and derived lipids. And in case of cell cycle and cell division, it is a third chapter. In case of cell cycle, cell division, we are observed two types of cell divisions. One is the mitotic division and meiotic division. In case of both mitotic division and meiotic division is mainly followed by that is interphase. Interphase mainly contain 3 stages: G1, S, and G2 phases. Then after mitotic division again contain 4 stages: prophase, metaphase, anaphase, and telophase. This is and finally cytokinesis. Result of cytokinesis, 2 daughter cells are formed in mitosis. In case of meiosis, we are observed same stages: prophase, metaphase, anaphase, and telophase. Then cytokinesis 2. Prophase, sorry, what is that? Prophase 1 of meiosis 2. What will observe? Sorry, meiosis 1 observed different type of stages. Prophase 1. So leptotin, zygotin, packaging, diplotin, and diakinesis in meiosis 1. Then after metaphase 1, anaphase 1, and telophase 1. In case of meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. Result of cytokinesis, we are observed 4 haploid daughter cells, 4 haploid daughter cells. So one more thing, after this unit, cell structure and function unit is completed. Then after only one unit is remaining there, that is plant ecology. Plant ecology is the last unit for you. Then after completion of ecology, plant ecology, we are completed all chapters in first year. Totally syllabus is completed after ecology ecology unit.
Kashinath sir, class is over.
Madam?
Class completed.